Hello, everybody. Welcome to the third installment of the OCB webinar series. We are very happy to have you. I hope you're filling out the poll as you join us and telling us where you're from. For those who don't know me, my name is Heather Benway. I'm the Executive Officer of the OCB Program and Project Office. I'd also like to introduce and acknowledge my co-host, May Mahegan, who's the OCB Communications Officer, and OCB's Administrative Associate, Mary Zawoski, as well as the OCB Scientific Steering Committee. You can learn more about them on our website in the About pull down. Today, we're joined by two members of the Scientific Steering Committee serving as co-chairs for this webinar, Jamie Palter from University of Rhode Island and Margie Friedrichs from Virginia Institute of Marine Science. We have, as of the time I started the webinar, we had 227 people registered. So that's pretty exciting. And we have well over 100 attendees already on the line here. So it's a really nice turnout. We're really excited about that. For those who don't know, the OCB Project Office is based at the Woods Hole Oceanographic Institution, and we receive generous support from NSF and NASA. OCB is a network of scientists working across disciplines to understand the ocean's role in the global carbon cycle and how marine ecosystems and biogeochemical cycles are responding to environmental change. This biweekly seminar series will really span OCB's overarching science areas, which you can see here in these green icons. And you can visit our website to find out more about our more specific research priorities. We usually, for those who don't know, we have an annual summer workshop in Woods Hole. It's a really broad science workshop that spans all of these disciplines and really draws a broad faction of the OCB community. And it's a great interdisciplinary meeting. And it's a really fun way to network and share new science. And this year, because of COVID-19, we were not able to have that. So we decided to try to have a webinar series to bring members of the community together virtually to share great new science. So we solicited ideas and abstracts from you as our community a couple months ago, and then the SSC and I went through submissions and we put together this topical webinar series. And if it's consistently well attended and well received, we might continue beyond the summer. I really hope that that can happen. So please share this with your friends and colleagues. So far, so good. We've had great turnout. You can find the schedule and details of the seminar series on our website. This will give you a sneak peek of the next two that are coming up over for the next month. The remainder of the webinars will have topical themes with 2 speakers per webinar, and you can, if you can't make it to any of these webinars, we do record them and they will be posted on our YouTube channel. And we always share the registration links for the webinar at the URL listed here and also via our e-newsletter and on Twitter. So hopefully you are able to learn more. You can always email us too. And that brings me to how you can get involved. If you're not already a member of the OCB community, we hope that you would like to become one. Our website and our e-newsletter information is listed here. You can actually subscribe to the e-newsletter just by visiting the website and scrolling to the bottom of the page, and there's a link right there to subscribe. You can follow us on social media. We're on Twitter and YouTube, and we also encourage you to live tweet during these talks. You can use the hashtag OCB webinars. And if you have any questions, please email us at the email at the bottom of the screen. Just a few guidelines. All attendees, there are many of you, so we have to mute you for the webinars just to reduce background noise. We ask that you save questions for after the talks. We'll have a brief Q&A after each talk. We have a Q&A function actually in Webex. This is the platform that we're using. You can access that via, there's a ... If you mouse over the bottom of your screen, you'll see a mute button, and you'll see a ... and that's where you can access the Q&A. So after the talk, the moderator will look in that Q&A section for any questions that you may have for the speakers. If we can't get through all the questions, we will be saving the Q&A and the chat logs from the webinar, and we will send any unanswered questions to the speaker along with your information so that speakers can get in contact with you afterwards. If you have any technical issues, feel free to use the chat function. And there is phone dial-in information if you have trouble with your internet and cannot keep the video going. You can always go back to the recording later. Here's a screenshot of what you're looking at right now, the Webex. You can see you can leave the webinar with the red X. The Q&A is under this ... The chat is under this icon here that looks like a bubble, a speaking bubble. And if you want to change the layout that you're looking at, the visual layout, you can do that in the upper right corner, and call in information. If you decide that your video is going wonky, you can use this pull down here with the circle to get details for calling in on the phone. So the theme of today's webinar is physical drivers of marine biogeochemistry and ecology. We're thrilled to have had so many people register. Today's speakers are Mara Freilich from the Massachusetts Institute of Technology and Woods Hole Oceanographic Institution's Joint Program in Oceanography, and Natalie Freeman from University of Colorado at Boulder. As I said before, we're also joined by OCB Scientific Steering Committee members Margie Friedrichs from VIMS and Jamie Palter from University of Rhode Island, who will be helping co-chair the session. I'm going to let Margie introduce our first speaker, Mara, while we transfer speaker role to Mara.
Okay, great. So thanks everyone for joining us today. It's my pleasure to introduce Mara Freilich as our first seminar speaker. Mara is a PhD candidate in the MIT-WHOI Joint Program in Amala Mahadevan's research group. Her research focuses on understanding the links between biological and physical oceanographic processes, and she uses a range of methods from theory and numerical models to observational work at sea. So before her PhD, Mara received her undergraduate degree in applied math at Brown University and completed a Fulbright fellowship in Santiago, Chile, where she was studying mathematical ecology. And she enjoys being on the water in boats of all sizes, from kayaks and tall ships to research vessels. So take it away, Mara.
Thank you so much for organizing the webinar series, and thank you to the OCB Project Office as well for organizing this, and to everybody here for attending. Yeah, so I'll be talking about the ways that the phytoplankton community structure affects fluxes of biogeochemical tracers, including carbon, oxygen, and nutrients. And so there's this diagram that I'm showing here shows two of the mechanisms of the biological carbon pump. And we'll focus a lot of this talk on thinking about carbon export. So one mechanism is sinking of particles. So this would be larger cells, for example, diatoms. Another mechanism that's increasingly recognized as important is a physically driven mechanism. It sometimes goes by the name of eddy-driven subduction, where smaller cells are swept to depth on currents at submesoscales or mesoscales. For both of these mechanisms, there's increasing recognition that the biological diversity plays a key role in the strength of the biological pump. For example, in the sinking flux, diatom diversity could be an important controlling factor on the export. And so what I'll be focusing on is the physically driven export, this of smaller cells and smaller size classes of non-sinking size classes. In either case, the flux is, well, think of a flux like an eddy covariance flux as a vertical velocity anomaly multiplied by a tracer anomaly. And so in the sinking flux, the vertical velocity anomaly is driven by the biological dynamics. So if the cells are larger, then they might be sinking faster. In the case of the physically driven or eddy-driven subduction, the vertical velocity is driven by the physics. However, the tracer anomaly is still driven by the biology. And so if we want to understand the total export, we have to understand both components and, in particular, the covariance between them. And so in the first part of my talk, I'm going to talk more about how the vertical tracer fluxes are controlled by biological communities and by the biological rates. And then in the second part, I'm going to take this theory and background from modeling to observations in the Western Mediterranean to ask how frontal dynamics modify the phytoplankton community composition and tie it back to the implications for export. And this talk is driven by the hypothesis that both physical and ecological processes generate diversity at the submesoscales, which can affect biogeochemical fluxes. And I hope you'll understand this better and what I mean by this throughout the talk. But to start out, I'm going to focus on this first branch, how phytoplankton community structure affects biogeochemical fluxes. And here I'm going to take a very simplified view of biological community structure. We say that community structure is encapsulated in its function, and its function is a net rate. And one reason that this is important is that net rates vary over a few orders of magnitude in the ocean, and the net rate of biological processes, the growth rate, for example, depends on the community composition. And so what are the implications of these different biological rates, particularly for a nutrient or carbon flux? And so what I'm showing here in this animation on the left-hand side, you have these two particles that are in a vertically oscillating flow field. So they're oscillating vertically in a flow field. One, the biological rate is fast. So there's fast restoring to an equilibrium concentration. And that's this particle on the left-hand side. And that means that at any moment, the tracer is nearly in equilibrium, and so has a very low anomaly. By contrast, with a slow rate, this particle is maintaining a nearly equal concentration throughout its trajectory. And so at the extremes of the trajectory now has a very large deviation from equilibrium. The tracer just doesn't have time to equilibrate before it's moved to a new concentration. And so that was looking at the tracer anomaly. Now a flux has to do with the covariance between the vertical velocity and the tracer anomaly. And so on the left-hand side, I'm plotting the flux, W prime C prime, as a function of the biological rate, lambda. We see that each curve has a maximum. And so each curve represents a different vertical velocity oscillation frequency. Now we're encapsulating the vertical velocity with this sine function that has a frequency omega. So with a slow omega, so a slow oscillation of those particles, we see the biological rate that maximizes the flux is relatively slow, whereas as the vertical velocity frequency increases, the biological rate that maximizes the flux increases. We can derive mathematically that for a given vertical velocity frequency, the flux is maximized by a matching growth rate. And so to understand this a little bit more, and to put it in a more oceanographic context, I'm going to use a 3D numerical model of a process study model of a mid-latitude eddy field. And so on the left-hand side of my screen, I'm just showing a snapshot of the vertical velocity at one depth that's within the nutricline or an oxycline in this case. And on the right-hand side, I'm showing a frequency spectrum of the vertical velocity. And what I want you to take away from this is that the vertical velocity has a full range of frequencies. So we'll couple this to a very simple biological model that's restoring, similar to the animation I showed you. And with a fast biological rate, now with this tracer that's embedded in the flow field, we see that there's a very low, or that the tracer anomaly matches the vertical velocity at any given time point. So that's this middle panel. Left-hand panel, I still have the vertical velocity. The middle panel has the tracer anomaly. And then on the right panel, I'm showing the flux, covariance of the vertical velocity and tracer anomaly. We see that it's positive everywhere. Now, by contrast, with the slow biological rate, we have a large tracer anomaly. Now, the tracer anomaly is 2 orders of magnitude larger than it was with the fast biological rate, but it's no longer in phase with the vertical velocity in the same way. And the result of it not being in phase, if we look at the bottom right panel, is that there are areas of positive flux and there are areas of negative flux. And so if the uptake rate is too slow for the vertical velocity, these areas of positive and negative flux cancel, because these anomalies are large, but they're no longer in phase with the vertical velocity. And that can result in a very small integrated flux, even though fluxes at a point can be much larger than with a fast uptake rate. And so the uptake rate that maximizes the flux now is just going to be an intermediate rate between fast, where the anomalies are small, but the vertical velocity and tracer anomalies are in phase, and slow, where the vertical velocity and tracer anomalies are no longer in phase, despite anomalies that are larger. And so if we integrate in this realistic ocean model, we find that as predicted by the one-dimensional theory, in this three-dimensional model, there's a particular biological rate that maximizes the flux. And so this is integrating over this full range of physical timescales, the full range of vertical velocity timescales. We find is that the timescale that's most important is around 3 days, which is similar to submesoscale timescales. And so in the ocean, the larger scales we think of as mesoscales, and those are eddies that persist on timescales of months. Whereas the submesoscales are much smaller scales, so 1 to 10 kilometers, and these persist on timescales of days or a week longest. And it's these fast fluctuations that are really dominating the interaction between biological rates and fluxes. And so that I was talking about the physical dynamics, but just looking at kind of one net rate per community. And when I transition into the observations, I'm going to be talking about ecological interactions. And the reason for that is that observations show that both ecological and physical factors are really important for contributing to diversity during a bloom. This is an example from California, where they've found that during a bloom, although the bloom was initially diatoms, the Pseudonychia, later on in the bloom, there was this very clear succession of species that included smaller cells, and that was driven much more biological dynamics than any of the physical factors like nutrients, temperature, or light availability. And so that returns us to this hypothesis that both physical and ecological processes generate diversity at the submesoscales. So I showed that this diversity could affect biogeochemical fluxes. And now I'm going to talk about this other branch from observations. How do biogeochemical fluxes affect phytoplankton community structure? And to do this, I'll be using observations from the Western Mediterranean, where in the Western Mediterranean, we have strong fronts where cool, fresh water from the Atlantic flows in and meets warmer resident Mediterranean water. And I'll be focusing on these two features I've highlighted: an anticyclonic gyre on scales of 100 kilometers in the west of this image, and then a smaller anticyclonic eddy, and taking observations at high spatial resolution in both. And in this image, you see variability in the chlorophyll at some mesoscales at these 1 to 5 kilometer scales, suggesting that there's interaction between physical and ecological processes here. And the reason we focus our sampling on fronts is that exchange between the surface and interior occurs at fronts. What I'm showing here is a process study model from the Western Mediterranean, and each dot is a location where a water parcel left the mix layer and went into the interior. And at each of these, and so these locations are kind of along fronts, so along these areas where we have higher density gradients, but they're kind of patchy along fronts. And we found that they were really driven by some mesoscale dynamics. And so this has been found globally, that this exchange between surface and ocean interior occurs at fronts. And the dynamics present here are mesoscale and submesoscale fronts, eddies, and frontogenesis. But across other dynamics, in mixed layer eddies, upwelling systems, and wind-driven systems, this exchange between the surface and interior really occurs at fronts. And Ruiz et al. In the Western Mediterranean encapsulated this by saying that 80% of the flux occurs in just 15% of the area. And so that's why fronts are particularly important, and why the ecological communities at fronts now will be particularly important. And so for my observational methods, I use flow cytometry to get quantitative cell counts of Prochlorococcus, Senecacoccus, and pico-eukaryotes. And I combined that with genomic analysis, with 16S amplicon sequencing, to get relative abundances of bacteria and photosynthetic eukaryotes, but I can also combine this with the flow cytometry to get absolute abundances of Prochlorococcus and Senecacoccus, which have 1 copy and 2 copies of the 16S gene respectively. So we get absolute abundances with very high phylogenetic diversity. And so to introduce these types, the Prochlorococcus and Senecacoccus are both cyanobacteria. They're both globally distributed, although Prochlorococcus's range is limited by temperature. And each, when we look at their diversity, they have ecotypes that are specializing in light niches. One key difference between them is that Senecacoccus uses nitrate, whereas Prochlorococcus mostly uses ammonium, although there are types that can use nitrate. And these are both very small, so they're in the small non-sinking size class. And when we take these cross-front transects in the observations, the transect that I'm showing here on the left-hand side now is a depth-distance transect across a front, with the dense side of the front on the left-hand side and the light side of the front on the right-hand side. What we see are these filaments of surface water that are transported to depth. And in this case, I'm showing apparent oxygen utilization, AOU, where high AOU means low oxygen concentration, so a lot of the oxygen has been used. And on this filament, and I'm pointing to with the arrow, we see this low AOU water being transported from the dense side of the front over to the light side of the front at depth. And we see this really rich submesoscale variability in these biogeochemical tracers. The question is, how is this driven by the ecological communities? And what are the implications of this for the ecological communities? And so at fronts of water masses. So on the right-hand side, I'm showing a temperature salinity diagram, and I'll show a few more of these. Point out that the Atlantic water, as I said before, is fresher and cooler, whereas the Mediterranean water is warmer and saltier. And so to interpret these observations and really disentangle these pathways, I pointed to this cross-front pathway, but I want to show this image again to really make the point that the along front flow is faster than the cross-front flow. And these are really three-dimensional features, particularly at these scales. And so even though I'm going to be showing these cross-front sections, we really have to think about these as 3D dynamics. Now returning to the temperature salinity diagram, fronts are meeting the water masses, but they're also biogeochemical fronts. So this is true here in the Mediterranean. Natalie will be talking about this next in the Southern Ocean. And one of the implications of that is that the communities at fronts could be different, and fronts are also locations where you can have nutrient supply to these distinct water masses. And so looking at this image, these horizontal or these diagonal lines now are density surfaces. And so looking along a particular density surface, we see that the Mediterranean water is more depleted in nitrate than the Atlantic water. And these profiles that I'm showing here are from that small anticyclonic eddy near the coast of Spain. And here we captured this mixed water mass that had an intermediate and it also had an intermediate nutrient concentration. Look at the biological response to this mixing. So this mixing was generated by the submesoscale intrusion that I showed you and occurred over timescales of days. We see that there's an active ecological response. So the chlorophyll fluorescence in this mixed water mass was much higher than anywhere else that we sampled within this eddy, except at the very surface in the Atlantic water mass, which is the other bright spot in chlorophyll. This mixed water mass, it did have a mixed community. So that was true across all the different groups that we sequenced, but I'm going to show this with Ostriococcus, which really exemplified this. Ostriococcus is another small cell. It's about this, it's a eukaryote, but about the same size as Senecacoccus. And it has, and we found two ecotypes here. We'll just call them O1 and O2. in the Atlantic water, almost all of the amplicons were of the O1 type, whereas in the Mediterranean water, almost all the amplicons were of the O2 type. But in this mixed water mass, we found a mixed community where both ecotypes coexist. And so this is very rare. The only place that these two ecotypes have been seen to coexist before are also in highly energetic regions. So due to mixing of water masses, like in the Kuroshio or the Gulf Stream. And so what we saw with this mixing induced by the submesoscales was an active ecological response to this nutrient injection. So it formed a distinct community at the front due to mixing, but there was also growth and shift in the community structure that could have implications propagating downstream. And one reason that this mixing is particularly interesting is because it's a density class that we did see in that subducted intrusion subsurface. And so these are communities that could potentially be exported along the filaments. And so I'm going to return to this cross-front transect and point out one other feature that I didn't point out before, and this is the submesoscale upwelling. So there's this small feature now that I'm pointing to with an arrow where we saw upwelling of high AOU water. It was depleted in oxygen but enriched in nitrate. And when we looked at the community along in a CTD cast right at the edge of that filament, we see that nutrient injection, so that's the purple profile on the plot on the right-hand side, which again is depth, and then the bars are showing the cell abundance of these cyanobacteria. And so we didn't see a numerical response in eukaryotes, but we did see a numerical response in the cyanobacteria, where it seems like the Senecacoccus are responding to this submesoscale nutrient injection that occurred over depth scales of 20 to 30 meters and over spatial scales of just a few kilometers. We don't see Prochlorococcus responding. This makes sense because Prochlorococcus couldn't use this nitrate. And so this is showing that these small cells could potentially be important for their responses to the submesoscale and then later for export. Here's my final example from this data set where I'm showing the impact of all of this for transport to depth and for export. And so here's a case where we sampled along what here I'm showing in the background a temperature intrusion. So this is another depth plot with distance along the transect. And we saw a temperature intrusion moving downwards. It was also had low AOU, high chloride, and it warm. And it was along a density surface. And so we expect this sort of subduction to be along a density surface. And so to the extent possible, we sampled across all of those characteristics, including preferring to be along a density surface. And what we saw is that as this filament moved downward, so from 30 meters to 180 meters, the community remained largely intact. And it was composed of Senecacoccus and this high light Prochlorococcus ecotype. And so this is decidedly a surface community now that was present at depths of 180 meters. And we expected that this subduction occurred on timescales of just a few days. And it's particularly striking if we contrast it with the communities that are normally present at that depth, which are indicated by the squares in this lower plot, which had either just lower cell abundance or even presence of the low light Prochlorococcus ecotype, which is what we would expect to be able to grow at these depths. And so as this filament moves downwards, its AOU also increases. So oxygen is either mixing with the surrounding water, or we're seeing a remineralization of the organic matter in this water mass and consuming oxygen. And we were able to measure respiration rates using incubation experiments with continuous oxygen measurements. And these respiration rates varied over an order of magnitude. And so knowing which of these respiration rates is most important or was the dominant one during the subduction event will really tell us how long do we expect this organic carbon to persist at depth, and really what is the implication of these whole communities, these whole surface communities being carried to depth effectively. And so this is beginning to tell us of what are the ecological dynamics within these downwelling filaments, and particularly how are these surface communities persisting at depth. And so what we found is that there's a tight link between the phytoplankton community structure and biogeochemical fluxes. And so from the model, we were able to show that biogeochemical fluxes are determined by the interactions between both physical and biological processes. In the model and observations, we said that ecological interactions are determining the responses to physical fluxes, so particularly in that mixed water mass. And finally, we see that small size classes contribute to export by responding dynamically to both upwelling and subduction. And going forward, we would really like to make the link in the observations back between the phytoplankton community structure and the biogeochemical fluxes. And this is all a work in progress. And I would love to hear any questions you have now or later. I have my email at the bottom. And I'll just end with acknowledgements of the larger number of people who made this work possible. And I particularly want to point out to my advisor and research group, Amala Mahadevan, and Alexandra Warden, in whose lab I did the biological data analysis. So thank you, and look forward to your questions.
Great, thank you so much, Mara. We might have time for a couple quick questions. So you can go ahead, anyone can go ahead and enter questions in the Q&A box, if there are any. And while we wait, I have one question for you, Mara. So you pointed to one biological rate that maximizes the flux, but there's some variability around that value, right? So what drives that variability?
Yeah, so that was going back to our model results, where I showed in the model that there was one biological rate that's optimizing the fluxes there. And I came to that result using Lagrangian particles. And so if we look at the some Lagrangian particles actually had different maximum rates. We find the Lagrangian particles that originate within fronts. So within these areas with faster vertical motion, they have faster biological rates that optimizes the flux, whereas particles that started within the centers of eddies or more quiescent regions have slower rates. And so we do see that potentially these fluxes could be affecting the biological community in terms of which community is able to optimize fluxes and use the nutrients most efficiently.
Okay, we have a couple questions here. I think, we've got a bunch of it. So I think that you're going to have to follow up with some of these folks afterwards, but we will go with the first name, Eric Rem. Do you think the community composition changes with depth along front with depth could be diagnosed optically from floats and gliders, for example, multi-wavelength excitation, chlorophyll emission fluorimeter?
Yeah, so I think there's a lot of potential for using optical measurements, particularly at these submeso-scale spatial scales. I think one of the things that was unique about this data set is that we were able to sample at this kilometer resolution using the CTD. But the other tool we were using, which is what gave us those high resolution transects of oxygen, something we called an eco-CTD, which is a toad profiler developed by Matthew Dever in our lab that has some of these bio-optical instruments at high resolution. And I think that's really the direction that will be really helpful for looking at the coupling at these spatial scales that are really small spatial scales, but also really fast. And so we need to be able to measure as synoptically as possible to capture the interactions with the submesoscales.
Great. Okay, so there's some other great questions here. I think hopefully you can follow up with those afterwards. But I think at this point I should turn this over to Jamie Poulter to introduce our next speaker.
Great, thanks.
Thanks, that was great. I really enjoyed it. So our second speaker is Natalie Freeman, who we asked the speakers to give us a little personal information, so we felt like we get to know each other even though we're far apart. And so she has a great story that she was growing up doing dancing and performing in musical theater, on track to be a professional dancer, not a scientist. She came across a National Geographic article on climate change and her world changed. She started doing all her class projects on climate change and her interest just took off. Now she's Dr. Freeman and she's currently a postdoc at the University of Colorado Boulder, where her research combines a suite of in situ and satellite data and model output to investigate the intersection of physical and biological oceanography in the Southern Ocean. Today, she'll be highlighting some of the work she completed during her previous postdoc at Scripps Institution of Oceanography as the Scripps Institutional Postdoctoral Fellow, and that she started during her PhD at Colorado Boulder as a National Science Foundation Graduate Research Fellow. And when she's not doing science, Natalie can be found spending time outdoors and tending to too many houseplants. And also, as we all are, dreaming of post-pandemic days that she'll be spending swing dancing again. So with that, I'm excited to turn it over to Natalie for a talk about the Southern Ocean.
Thanks, Jamie. Hi, everyone. Thanks so much for tuning in. I'm going to follow Mara's really nice overview on these small-scale features and zoom out and talk about the intersection of physics and biogeochemistry at much larger scales. A lot of the work I'll be showing today has been, I should ask, can you see my cursor? Can someone say?
I can.
Okay.
Yes.
So has been done in collaboration with these fantastic scientists. And the title here refers to specific work that I'll touch on a bit later that's currently being written up for potential publication in GRL. So for the next 19 minutes, I get to, thanks to the OCB team, I get to talk about my favorite ocean, the Southern Ocean. And for those of you that aren't familiar with this part of the world, I want you to know that the Southern Ocean is unique and important. It's the only place on Earth where waters circle the globe, and this is through the opening of Drake Passage between South America and Antarctica. And here I'm showing you a vertical slice of the ocean in Drake Passage with Antarctica now on your left. And the colors here just represent the density structure where we have these dense bottom waters and these more light surface waters here. Now the westerly winds drive the largest current on the Earth, the Antarctic Circumpolar Current, or ACC. And these winds also drive an overturning circulation, bringing up old, deep waters that are rich in carbon and nutrients to the surface. And this helps fuel biological productivity, and it also promotes the exchange of heat and carbon with the atmosphere. And from here, these waters get transformed. So they either form dense bottom waters, or they travel out of the Southern Ocean as mode or intermediate waters. And this is what gives the Southern Ocean its unique ability to impact the rest of the global ocean. So it's not just this isolated, faraway basin. It has the ability to be affected by and affect the rest of the global ocean. Now, the ACC is made up of many fronts, and like Mara introduced, fronts are features with strong lateral gradients and properties like temperature, salinity, and nutrients. The one difference between Mara's fronts that she discussed is that these are far more deep-reaching due to this vertical circulation. Hers were much more shallow. And one particular front I apologize if you hear construction, of course, right now, next to the apartment next to me, they're starting works because someone just moved out. So I'm sorry if you hear that. So the front I'm mostly going to speak on today is the Antarctic Polar Front, depicted here by this black contour. And this particular front is exhibits sharp gradients in temperature and salinity and also nutrients. And it separates unique physical and biogeochemical zones, so between the Antarctic zone and the polar frontal zone. And it also separates unique temperature-dominated regime to the north of it and a more salinity-dominated regime to its south. And so as a result of this physical circulation, we can see here the resulting nutrient distributions at the surface. So I'm showing you here the surface silicate concentration and nitrate concentration taken from the World Ocean Atlas database in millimoles per cubic meter. And we can see that around Antarctica, that most of the surface waters are filled with high concentrations of silicate and nitrate at the surface. And this again is tied to that underlying overturning circulation. And what we can already notice here too is that we can see the presence of a silicate front. So right where I'm drawing my mouse, you can see that these are the sharpest gradients, north-south gradients in silicate. And if I were to plot over this, the average location of the polar front, which is typically more defined in temperature, in the literature that these would roughly coincide with one another. And the presence of the silicate front is actually largely understood to be tied to that of the polar front, given these underlying physics that set them up. And so throughout the literature, these two fronts, the polar front and silicate front, are often used interchangeably. Now, our current understanding of these nutrient distributions at the surface is actually hinged on quite sparse ship observations. So here in the middle, I'm showing you the number of silicate observations and nitrate observations that actually went into making these pretty plots. And mostly if it's grayscale, there haven't been very many observations, maybe 2 or 3 in the historical record. But where we see color, we can see where we tend to sample or repeat our observational transects, like in the Atlantic sector and south of Australia. And I wonder why. This is just an example of what it's like to go out to the Southern Ocean and try and sample. And this is from a trip that I took back in 2016. So this is the reason for such sparse observations. So why do I bring this up? Why does our confidence in our nutrient observations matter so much? There are many reasons to that question, but I want to focus on one. Particularly that the silicate distribution in the Southern Ocean is important in terms of the biogeography of the Southern Ocean. And what I mean by that is the large-scale geographic distribution of the dominant phytoplankton that live here. So the who lives where. And so what we see here are the two illustrations of the two dominant phytoplankton species, the coccolithophore and the diatom. The coccolithophore is a calcifier. It builds a shell out of calcium carbonate or chalk, and the larger diatom is a silicifier and builds its shell out of biogenic silica or opal. And while both of these phytoplankton require nitrate and iron and other nutrients to grow and thrive, the distinction here is that the diatom is the only phytoplankton in this image that requires and uses silicate. So it's the only one of these two that actually cares that we have all this silicate-rich water south of the silicate front. North of the silicate front, diatoms are more likely to experience silica limitation. So as you move northward, you're getting into waters with less than maybe 5 millimole per cubic meter of silicate available. And we do document this in situ out in the open ocean, where we find that the ratio of silicate to nitrate availability is actually a major driver in the partitioning between these two species. And this is particularly observed at the location of the silicate front. Now, this front biogeography relationship can be seen from ships, but really cool thing I want to just plug really quick is that we can also see this relationship from space. So Barney Balch's work on developing a satellite-based proxy for the presence of coccolithophores through an empirical estimate of their shell concentration through the backscattering properties of their shells has really enlightened us. So here I'm showing you a snapshot from December 2002 of this NASA coccolithophore product, where warmer colors are showing us where more coccolithophores are in the water. And on top here, I plotted in white the location of the polar front in that month. And what you can probably have already guessed in your head is that, wow, look at the gradient in coccolithophore community across the front. So we see high concentration of coccolithophores north of the polar front, and then south of the front, not very many at all. And so while we know that the polar front is characterized by sharp gradients in temperature, salinity, also silicate, we can also see that it's also a gradient in coccolithophores. And so we could imagine that diatoms are what's dominating south of the polar front here. So if this intrigues you at all, I recommend you to look up Barney Balch's work. This is actually a circumpolar feature we can see around the Southern Ocean, and it's called the Great Calcite Belt. But I hope by now I've convinced you at least that the front and its location and its variability has the ability to impact local biogeography. And I'm just May will also drop in the chat some recent work that highlights more of these linkages. Okay, so you can probably draw this in your sleep by now, but I redrew in a schematic form this modern day biogeography setup where we have the partitioning between calcifiers north of the front and silicifiers south of the front, largely driven by this distribution of silicate across the front. Now, how surprised would you be if I were to tell you that this biogeography pattern is helpful to paleoceanographers to understand past climates? So in the sediments, they show us a very similar pattern. So as you move north, from north to south along the seafloor, you move from more chalk-dominated sediments to increasingly opal-dominated sediments, roughly mirroring the biogeography that we see at the surface. And so while I won't go into the methodological detail that they employ, but just know that they use the sedimentary makeup of chalk versus opal to reconstruct past locations of the polar front. And that then helps them get a better picture of what perhaps past levels of biological productivity were at the surface, and then back out what that could mean for the overall climate state on these glacial interglacial timescales. So some of their work hinges on how well we understand the modern day biogeography at the surface. This understanding also helps us make future projections. So recently we used a climate model to better understand silicate and silicate front variability, because we don't have very many silicate observations. And the CSM large ensemble predicts a more poleward silicate front in the future. So don't pay attention to much, but this black line, a decrease in latitude on average by about 3 degrees by 2100. So the silicate front shifts this much south on average. And this is associated with large-scale decreases in the surface silicate-to-nitrate availability that we discussed earlier. So we can imagine that it's a shrinking of the total square area of this this pocket of silicate-rich waters here, and therefore sort of reducing the square area that diatoms dominate and occupy and live and produce and all of that. I say here with impacts on biogeography unknown, because with this particular model setup, we weren't able to directly ask what happened to the coccolithophores and the diatoms with this decrease or this shift in the silicate front at the time. But I want to plug very quickly that this work will be possible in the future because Kristen Krumhart has been implementing a coccolithophore specific phytoplankton functional type into the CSM biogeochemical model. And she's already seeing quite realistic biogeography in the Southern Ocean. And I also want to say that she not only implements them into models, she illustrates them. She is the very talented illustrator that we've been seeing throughout this talk. So thanks, Kristen, for your work and your images. So to quickly circle back on the influence of the Southern Ocean physical circulation on the rest of the world, Sarmiento's work back in 2004 really demonstrated to us how any perturbation in the silicate-to-nitrate availability in the Southern Ocean really has the potential to impact what's going on outside of the Southern Ocean, in particular low-latitude productivity. And therefore, we could think of that as having impacts on air-sea carbon exchange and also then global temperatures and how much climate change we feel. And Keith Moore's recent work on Southern Ocean nutrient trapping beautifully demonstrates the Southern Ocean's influence on the rest of the world. By looking out to year 2300, he sees really dramatic changes in the nutrient dynamics in the Southern Ocean and goes into great detail about how this affects the rest of the world's biological productivity. So if you haven't checked that paper out, I really suggest that you seek it out. Now, this is the last thing that I'll say on this particular result. Another finding from this modeling study of ours was that we do find this shift in the silicate front, but it was actually independent of a shift in the polar front. The polar front didn't shift. So this sort of turns that paleo connection I mentioned on its head. So if the silicate front can shift and diverge from the location of the polar front by this much on these long timescales, perhaps our current understanding of this inherent coupling of the two of these fronts needs to be revisited. So how does this influence our ability to interpret the sedimentary record to get at these past locations of these fronts? So definitely something worth looking at in the future. So all that I've been telling you is really just to motivate this most recent work in the Drake Passage, where we're trying to better understand nutrient variability across fronts. And so we take advantage of the longest year-round biogeochemical time series in the Southern Ocean. Between South America and Antarctica, the Gould makes crossings year-round to resupply the Antarctic research stations. And during about 5 to 8 crossings per year, we sample for surface macronutrients like silicate, nitrate, and phosphate, and also temperature and salinity. And this includes wintertime transects, so this provides much needed year-round information on these biogeochemical properties. And because aboard the Gould, the Scripps high-resolution XPT program is being conducted, we also have high-resolution temperature data in order to co-locate where the fronts were during each crossing. And so everywhere we have a Drake Passage time series or DPT sample, which is these little circles in space, we can also analyze those values in the context of fronts and frontal zones, which are the colors here. So for instance, this particular sample, we know that was representative of the Antarctic zone, this light blue color. So with this, we were able to provide a first estimate of nutrient distribution and variability across these fronts on seasonal timescales in our most recent JGR Oceans paper. And I won't be going into much of this today, but it does go into great detail of what we've learned so far from this really novel data set. In particular, just how uniquely different silicate and nitrate are, particularly as you move more and more south. And so I suggest you read this paper, even if especially if you're a biogeochemical modeler, maybe test and see how well your nutrient cycles are doing. But the one thing I really want to focus here today from the Drake Passage time series is the interannual variability and change from 2004 to 2018 in these nutrients. And there's a lot going on in this figure. I just want you to focus on the darkest blue line of the monthly time series of silicate and nitrate in micromoles per kilogram. And the darkest blue colors are indicating that we've taken these samples from the southernmost zone in the Drake Passage, south of the southern ACC front. And if we follow this darkest blue line in time, we see that in 2016, silicate jumps and it exceeds background mean and variability levels. So near 80 units, where normally it's wiggling around 40 or 50 units. And with the nitrate, we see there isn't that dramatic of a signal. So silicate's really standing out here. And so this begs the question, what physical and or biological processes could have driven the 2016 silicate event? And in the paper, we go through some of the plausible explanations, but I'm only going to highlight 2 here. So the ENSO and southern annular mode are 2 leading modes of climate variability that can imprint on southern ocean physics and biogeochemistry. In general, a positive phase in ENSO in the gray, it also called an El Nino, typically occurs during a negative phase of the SAM. A negative phase of the SAM could be explained as a weaker westerly wind belt. And so from 2004, we see that in 2015, that one of the 3 largest ENSOs ever recorded, El Ninos ever recorded on record, occurred against a backdrop of an already really strongly positive SAM. And this doesn't happen very often. They're generally out of phase. And we know that just soon after that, we saw that increase in silicate-to-nitrate availability in the southern Drake Passage. And so this convergence of these 2 positive modes resulted in a strong, sustained high pressure anomaly that moved through the Drake Passage region, indicated here by these solid contours, and I plotted them over upwelling anomalies, so reds being anomalous upwelling and blues being anomalous downwelling. And so this atmospheric pattern resulting from the SAM-ENSO event can try to help us infer what might have happened, that we'd see an increase in silicate, but not in nitrate and phosphate. And so first, we can see that anomalous upwelling along the Antarctic coast could have driven up more silicate-rich water from depth and propagated that signal through the region. And also, we can see through this progression that the winds, the strength and orientation of the winds more and more parallel to the peninsula suggests that there was increased offshore transport of shelf waters, which could have supplied the Southern Drake Passage with a sedimentary source of silicate, which could help explain that this would be independent of seeing anything in nitrate and phosphate. So sort of follow-up questions that come to mind is, can we expect such dramatic nutrient dynamics with more common occurrences of these positive in-phase events in the future, like Wang and Kai's work suggests? And would we be able to detect this silica anomaly in low-latitude mode water? So if we went out, could we actually see the 2016 silicate event? That would be interesting. And I'm running out of time, but I just want to highlight the importance of prioritizing silicate observations. I've just demonstrated to you that through the concurrent sampling of both silicate and nitrate, we were able to detect a physical and biogeochemical linkage with a large-scale climate forcing that otherwise would have gone undetected if we had just been sampling for nitrate or phosphate. So nitrate and phosphate alone couldn't have revealed one of the 3 largest ENSO events on record and how that might have influenced the Southern Ocean. And I'll end there. Thank you so much.
Great. That was great. Thank you so much. If people want to type questions into the question bar, we have time to go through a few. And I can ask you one while we wait to see if it come in. I don't see any yet. One thing I was curious about is there's been a lot of work showing that the fronts of the ACC can be like leaky at hot spots of exchange that might be topographically driven. I'm curious if you can see the influence of that leakiness on community composition. Has anyone looked at that at these hot spots of exchange?
That's a good question. I haven't looked at it, and nothing's coming to mind that someone's done that. Maybe Mara's heard, but that would be a really cool study.
Put that together. Anyone else have anything? I don't see anything in the Q&A. We see a lot of people saying thanks for the interesting talks.
Oh.
So Heather, should we? I'm not seeing new things coming in, but
I don't see anything yet. I'm watching the Q&A. I'm watching the chat. You might be off the hook, Natalie.
I'm happy to respond to emails or Twitter or anything like that.
Yes, we will be saving the logs here. So if anything is not answered yet, we can certainly follow up. All right. Well, with that, I'd like to thank everybody for joining us. It was a really interesting set of talks, and we look forward to hosting again in a couple of weeks. Again, if you have any questions, please contact us or the speakers. We're happy to answer them. One person said, thanks. Thank you for the wonderful webinar. Thanks. You should check CESM.
Large ensemble.
Large ensemble, both ENSO and SAM in phase to silicate to nitrate ratio. Okay.
Great. Thank you.
Yeah. Interesting. Heather, do you have the response from the polls? Are you able to release
that? Yes, let me see if I can share the poll response. I think I can. All right, can anybody?
Yeah.
That just
can you
came
see
up
it?
in the polling. Yep. 64% from North America.
Yay, we had somebody say Drake Passage.
Yeah, I know. I want to know who that was.
It wasn't me.
I just saw another question come in. I just want to check and make sure. Okay, they're mostly just thank you for the great talks. So
Thanks for tuning in.
thank you to our speakers and our SSC chairs for this great session, and we'll see you all in a couple of weeks. Thank you, everybody.
Bye. Thank you.
Heather, do you need anything from